My name is Summer Simone Chin, and this is Single Recessive Epistasis, Fazzini Boots, and Influencers. Before we talk about epistasis, let's talk about genes and what genes do. Commonly, we know genes as getting certain features such as our eyes from our mom and nose from our dad. Genes are understood by using alleles which code for genotypes. These genotypes are then used to code for proteins called enzymes to express what is called a phenotype. The phenotype is the appearance. For example, big B, big G, little g is the genotype and could code for proteins to express big green flowers. The big green flowers would be the phenotype. Epistasis is when two different genotypes can contribute to the same phenotype. For example, let's say you start with a red liquid and an orange liquid. These starting liquids represent genes. The red liquid is then mixed with the green liquid, and the orange liquid is then mixed with the white liquid. The green and white mixer liquids represent proteins. In the end, both mixtures appear to be yellow, representing the phenotype. In genetics, this is understood as epistasis, as the epistatic gene masks the actions of other genes, allowing multiple genotypes to lead to the same phenotype. In single recessive epistasis, the homozygous recessive allele, a, will prevent other alleles that have a, from expressing another phenotype. This then limits the number of phenotypes to 3 instead of 4. In this presentation, I'm going to use an example of boots and influencers to explain single recessive epistasis even better. There are 3 types of boots. We got plain work boots, black leather boots, and Fazzini boots. If the influencers have the homozygous recessive a alleles, they'll be stuck with plain work boots. If they have a big A allele, they'll be able to take enzyme A to level up to black leather boots. And if they have both the big A and big B allele, they'll move along both enzyme A and B and be afforded the luxury of the high fashion Fazzini boots. In this diagram, we can see that a only gets us to the plain boots. But if we have a big A, we can take enzyme A and get to the black boots. If we have little b, we're stuck at the black boots. The only way we can get to these Fazzini boots is if we have a big A, big B to take enzyme A and enzyme B to get to the Fazzini boots. In a dihybrid cross, we see a cross between 2 heterozygous dominant A and B alleles. This cross then results in 9 Fazzini boots, 3 black, 3 plain, and 1 plain. As we can see, the plain phenotype repeats since both homozygous recessive A alleles are in the formula. Therefore, it can be condensed to 4 plain, giving us a 9:3:4 ratio. This next cross is a different type of cross. It's not a dihybrid, and therefore the results of our progeny are different. This cross results in half Fazzini boots and half plain. This is the end of my presentation. This is my works cited for my images and my information. Thank you so much for your attention.